So I am going to try to be fast. And those who know me know I'm tempted to just speak very quickly. I'm going to try not to do that, and instead just cut down on the amount I say. But basically what I want to talk about is work that I started when I was a graduate student postdoc here and have continued since, which is looking sort of, what is it, looking, basically I want to understand how the environment affects individuals through physiology, and then how that translates up to affect populations and whole communities. And a lot of the work I did when I was here was trying to understand how temperature, for example, affects metabolic rate and physiological rates, how that couples with body size and affects populations, and then what I'm going to talk about today, mostly what I'm sort of working on a lot in my lab now, is how to try to go from populations up to communities. And really, it's a long way to go to communities, but at least try to begin to put in species interactions, which I feel like is one of the steps you need to go up to communities. But I think that's okay. And thanks to the previous couple of talks, I can just go through this very quickly. Thinking about mass and temperature, the mass specific metabolic rate from a lot of theory and data, we can say approximately goes like mass -1/4. This bolts in fact with temperature, this activation energy that Wenyu talked a little about. And the inverse of a rate is a time. Thinking about biological times is going with mass and temperature is just the inverse of that. And if we look at, for example, at population growth rate that's mass corrected versus 1/kT temperature, we get a slope of -0.68, which is very close to what you'd expect from metabolic theory of ecology. It's across lots of different taxa, unicellular eukaryotes, vertebrates, multicellular eukaryotes. And if we look at mortality rate, mass corrected mortality rate versus temperature, we also see a pretty strong correlation, and an activation energy that's a little lower, but still close to the range expected from metabolic theory of ecology. And this is a paper I published with Jim that Jim helped me a lot on. And I remember talking with Jim a lot about this figure in particular because these data are from the field. And really our model is all about how individuals or species in isolation, how their metabolic rate affects their activity or their velocity or their aging rate. But this really has to do with interactions, right? Because this has to do with fish being caught or eaten. It's not them dying from just natural causes. It's them dying from being caught and eaten. So it's surprising almost that it works so well when you have interactions with other species. And so the question is really, how does this work so well when you have interactions with other species? It seems like you shouldn't be able to figure that out just by focusing on one of the species involved in the interaction. And so that's why I've been trying to figure out more deeply by thinking about consumer resource dynamics. And if you think about sort of the simplest equations you can write down for consumer resource dynamics, these you can either Locke Volterra or MacArthur, different things like that. They all basically have forms similar to this, where this R is population growth rate and the Z is mortality rate, which are the two previous data slides I showed, so we have an idea how those scale already. But these two equations are coupled together. The N is the prey and the P is the predator. They're coupled together through this consumption rate term. And so the question is that consumption rate is sort of what's driving this interaction. So the question is, how does that consumption rate vary with body mass and body temperature? And using this model, which is actually very similar to, inspired by a model Walter Yates used for thinking about home range within species interactions, this is between species interactions, thinking about predator-prey, where sort of what determines, if you think about consumption rate, the two species have to encounter each other for attack and the consumption event to happen. And thinking about consumption rate has a lot to do with the velocity at which the predator and the prey move around, the detection distance at which the predator and prey can see each other, and also the dimensionality in which you're searching, which I'll say a little bit more about as much as I have time to. But that's related to sort of where you can find the prey, right? The prey, how many prey are around and where you can look for the prey affects the encounter rate also. And where you can look for the prey has to do with dimensionality. So that's the first bit, of size and dimensionality. Just to think about this a little bit more carefully, there are different types of consumer resource interactions. Active foraging, where both the predator and prey are moving, so both individuals' metabolic rates or activities are sort of important for the interaction. Sit and wait, where primarily what's moving is the prey, so the sort of prey's metabolic rate is driving that interaction more. And then grazing, where the prey, if it's grass, is still, and the predator is in this case a rhino or hippo. is moving around. It's primarily driven by the metabolic rate of the hippo. And so this has to do, so this also, besides just the foraging strategy used, the dimensionality you're searching in, if you're looking for on the surface of, say, a seabed or terrestrial on the ground, or aerially or in a water column, 3 dimensions, that can affect your ability to find prey and search as well. In a paper that my group just published, we basically shown if you divide by 2D versus 3D, in 2D you see scaling of consumption rates with consumer mass, and there's a whole other part to talk about with resource mass and size ratios I'm not going to have time to go into. You get that the scaling exponents in 2D for search rate is 0.68, and for consumption rate is about 0.85, so it's around 3/4. It's still a little higher than 3/4, but around 3/4. And in 3D, it's actually 1.041 and 1.06, so it's definitely higher than 3/4. And it's also not just higher than 3/4, it's higher than 1. And thinking about equations, if you have sort of terms that are sublinear versus superlinear, greater than 1 or less than 1, that can affect completely the dynamics that you see. And I'll say a little bit about that. But this is an interesting result. We're surprised to see it. And one caveat I should say here is that all these data are for pairwise interactions. So you just have one consumer and one resource. But in nature, you often have one consumer eating multiple types of resources. And that's sort of another piece that's still left to go in terms of the theory and data about communities. Here, using this theory, putting into the predator-prey equations I showed earlier, with that result for consumption rate, we can predict what size ratios, this is resource mass and consumer mass, what size ratios should be allowed before you have extinction of the consumer resource. And these are data from real communities. And what you see is that in 3 dimensions, you have a lot wider size ranges, a lot wider size ratios of consumer to resource. And the data match onto that pretty well. And so, for example, I can explain why things like whales can feed on such small organisms as krill and still be stable. And you notice there are some big, large regions that are empty at the low end. And we think that's just because of a lack of sampling at the low end, but it'd be good to get more data there and to go that more deeply. And looking at 2D versus 3D, in 2D, thinking about the dynamics, like I said, if you have greater than 1 scaling versus less than 1 scaling, it can affect the dynamics. In 2D, what you have, the dynamics go to a single point, a fixed point, so you have a stable, sort of a more stable system. In 3D, you get these limit cycles, so you go into a stable limit cycle, but this actually creates more booms and bust dynamics. We're more likely to get a boom in your species abundance or a bust in your species abundance, which means you have larger fluctuations, which can make you more prone to extinction in these 3D environments, often aquatic. You get faster. Here we thought about Damon's rule today. Gives different predictions for Damon's rule in 2D versus 3D. And this exponent is actually wrong. In 2D ends up being closer to -3/4, and in 3D it's steeper. And before I finish, I have to say, but also the T to acknowledge Winyan's talk exactly. And so just to say one thing about the T, we've also been looking at how temperature dependence varies in species interactions. And this is a distribution for activation energies across a huge swath of data for interactions. Well, actually, this includes physiological traits and interactions. But if we look at just the interaction traits, the green here are positive traits, which means things like attack or consumption. And the red data here are traits that are like escape or defense, more negative versus positive traits. And the red traits are on the left side of the distribution, so lower activation energies, meaning less response to temperature. And the right-hand side is positive response. It means it's a stronger response to temperature. And this could be related to something like a thermal life dinner principle, that you have a stronger selection to save your life if you're the prey than you do to catch your dinner if you're the predator. So you're gonna, if you're the predator, respond more to the thermodynamics, but if you're the prey, you have more stronger evolutionary pressure to sort of overcome the thermodynamics and maintain your escape velocity to get away from the predator. And the last word I'll say, and then I'll say my things.
You've got 1 minute.
Okay, I'll work. Thanks. For video tracking experiments, I don't know if this loaded, if I can play this while I'm finishing. But no, I think the movie's not here. But there should be a movie here. But basically now we have these wingless fruit flies and spiders, and I have different types of spiders. And we can film it with video for hours, and then go in with software in there and track them, and track each individual, and get its velocity, turning angle, handling time for the interaction and being eaten, and actually even just from antenna movement, leg movements, to behavioral data, all kinds of data we can get from these predator-prey interactions now for several hours, which is, we're just now getting that data. It's pretty exciting. And here are examples just of like velocity profiles and turning, well, this is actually all velocity, different measures of velocity. But you can do all kinds of cool things with these data. And that ties into what Ethan talked about earlier, that I also have a great fondness for this macroscope of collecting lots of data on temperature and body size interactions, and actually video tracking now as well. So I'd love to, we're trying to get actually data to make public and put up on a website, so we should talk and link those together. And finally, I want to thank all of these people and say just a few words of thanks to Jim very quickly, which is that, I should say for these video tracking experiments, I just went out last week for the first time and caught the spiders myself, which was quite an experience given that I'm a physics PhD. And I want to say that just thanks to Jim for taking a chance on me, a physics PhD, someone who hadn't taken a biology course since high school, which was anatomy and physiology, so it's a big chance. And then I just want to say thanks for teaching me so much about the big picture and intuition for biology. I learned so much biology from you. And how to write a biology paper, which is different than a physics paper, and in my sense harder, but also better in terms of explaining things to the reader and engaging the reader. And finally, I just want to say the first time I ever talked to Jim on the telephone about coming, he told me that if I came, he would treat me, he treated everyone as a colleague and a collaborator. And most advisors don't say that. And I have to say, Jim not only said it, but he really meant it. And Jim has always treated me as a colleague and a collaborator with great respect and patience, and I really appreciate that. And I'm trying to learn how to be as good a mentor as that still. I'm still working on it. But thank you, Jim.